Hey everybody, in this video we are going to talk a little bit about dosage compensation. Okay, so where does this topic start? I think the best place, the most interesting place to start is what happens if we look at human somatic cells, so non-germline cells of people with different karyotypes. Okay, so let's say these are nuclei in different somatic cells. I think I need about 5 of these to get the point across. So now let's assume this is a nucleus from a somatic cell from an individual with this karyotype, 46XX, and you should watch the previous videos to know what this means, but essentially 46 chromosomes, 2 X chromosomes are in those 46. And then 46XY, 47XXY, 45X, and let's go with 47XXX. Now this is triplo X, and this is the Klinefelter karyotype. This is the Turner karyotype, things we talked about in the previous video. I don't think we talked about this one. Essentially, humans with 1 extra X chromosome are essentially no different than humans with 2 X chromosomes. There might be slight differences, but I'm not sure how well that has, if that's been confirmed. So, okay, so let's look at nuclei from somatic cells of these individuals. And this is, I think, back in the 60s where scientists were first noticing a difference. So when using certain stains, what they would see in nuclei of somatic cells, in this individual right here, they see a darkly staining body along the nuclear membrane. So this is a nucleus, this is the membrane. We're not looking at the cytoplasm, and they use certain dyes and they see a darkly staining structure along the membrane here. Now they don't see it over here in the 46XY, nuclei from 46XY individuals. They did see one here. They did not see one here. And in this individual here, they see two darkly staining bodies. And these are named after the scientists who discovered them. I think it was Murray, the first name Murray, but Murray Barr, and they're called Barr bodies. And it turns out that they are inactive X chromosomes. So cells in mammals, in humans and other mammals, possess a process called X chromosome inactivation, by which the number of X chromosomes are determined and all but 1 X chromosome is inactivated and is put into a Barr body, what's called a Barr body. And this inactive refers to most of the 1,000 or so genes. Most of them are inactive. We'll see towards the end of this video that it's important that at least one of them remains highly active. But for the most part, most of the genes are thought to be transcriptionally silent. So, okay, so that is a dosage compensation mechanism. And so what is dosage compensation? Why is it important? So, well, with respect to humans, right, we have XX humans have 2 X chromosomes. XY humans have 1 X chromosome. And it seems that of the thousands of genes, or I should say 1,000 or so genes on the X chromosome, it is important for most of those to be expressed equally, approximately equally, in XX and XY humans, so genetic females and genetic males. And without this dosage compensation mechanism, there'd be an imbalance. We'd have more of the X gene products in genetic females than we would in genetic males. And apparently that's bad. That's bad. So evolution has come up with a way to balance the expression of genes from the X chromosome in males and females. So in mammals, this dosage compensation works by inactivating an X. But in other organisms, such as flies, at least for Drosophila melanogaster, which is a model fly that's used for genetic research. So males have one X chromosome, females have two X chromosomes, but the genes on the X chromosome in male flies are expressed at twice the level compared to the level of expression from a single X chromosome in females. So by expressing or using a mechanism that increases the expression of genes on the X chromosome to twice the level, males are able to achieve the similar expression of X chromosome linked genes that is seen in females. So dosage compensation can be achieved, well, between sexes, can be achieved by a number of different mechanisms, but the version in mammals, the mechanism in mammals involves inactivating an X chromosome, so kind of tucking it away in that bar body. Now, so there are some interesting things going on here with X chromosome inactivation. And it started with a scientist named Mary Lyon, and I believe she is working in the 1960s, 1970s, and she proposes the Lyon hypothesis, which we can now call the Lyon model. And so in the Lyon model, this is for X chromosome inactivation in mammals, including humans. So all X chromosomes are active after conception. So in XX zygote, both of those X's are active. So a few weeks after, I don't want to give a time frame here, but I'll just say a few weeks because this time point would probably be different in mice and cats and humans. But at some point after conception, let's say a few weeks after conception, one X chromosome is chosen at random for inactivation, and it stays inactive. That same X chromosome stays inactive through replication and cell division. So once an X chromosome is chosen at random for inactivation, in all cells that are in the lineage of that cell where the X has been inactivated, that all daughter cells and descendants of that cell will keep that same X chromosome inactivated. So maybe I can diagram that. So here's a zygote. After a few weeks, we're going to have an embryo here. That's a terrible embryo here, right? But let's say we got a couple of million cells in here. And all the X chromosomes are active here. Let's say this is an XX zygote, so two X chromosomes. Now let's take one of these cells here. And we have an X chromosome here from mom, so X maternal, and there's an X chromosome that was from dad, inherited from dad here. This is X paternal, and they're both active. Now let's say this is, a few weeks after conception. So each of these cells at this time point, a few weeks after conception, each one of them is going to choose, but randomly, one of these X chromosomes to become inactivated. And let's say this cell right here happens to choose the XP as the one that is inactivated. So the paternal X, the X chromosome inherited from the father, is inactivated. Again, this is the nucleus of the cell, and this is the nuclear membrane. So that gets tucked away into the Barr body, that darkly staining region. If you use a certain stain, darkly staining region along the nuclear membrane. And the X maternal, that one, remains outside the Barr body and transcriptionally active. So if this should replicate, like it will, the DNA should be replicated and this should divide. Well, the daughter cells will have that XP still inactivated. So how that happens, and I'll talk, a lot of this process is not completely understood. We know it happens, but we don't really know how it happens. So how these get replicated and the same chromosome is placed into the Barr body each time after replication and cell division. And this is just one cell. So in other cells, the maternal one might be the one that's inactivated, and the paternal may be active. Again, that's a random process. That's a random process that determines which one is inactivated. But once it's inactivated, it stays inactive in all descendant cells. And there are examples of studies that have been done on this process and show that, yep, this is how it works in humans. But I think the more interesting ones involve, things we've probably all seen before. So cat coat color or fur color in cats, is a good model for studying or investigating the lion model. Not lion, the lion model. So cats, coat color in cats can be essentially either black or red. And this is probably something we've seen in the past, right? With eumelanin and pheomelanin in mice. So sort of a similar thing. But the coat color gene in cats is on the X chromosome. And one allele, we can say one allele is C for coat color, so the gene is named C, and the CB allele, let's say this allele is for black coat color. And this CR, this is for red coat color. And red is more of an orange, right? It's more of an orangish red. So this allele is for black pigment, making black pigment. This one is for making red pigment. Now, male cats, right, with XY chromosome composition, sex chromosome composition, can be either CR, right, the Y chromosome doesn't have the coat color gene, so males can typically only have one allele for the coat color gene, so CR, or CB. So this is going to be red cat or orange, and this one's going to be black. And we have like brown and gray, and so those are all different levels of pigment production that lead to those different shades of these colors. But males can be either a base color of red or a base color of black. Now females, typical females, 2X chromosomes, can be red, can be black, or they can be this sort of heterozygote right here. So homozygous for red, homozygous for black, or heterozygous for this genotype right here. So what is this? Is this cat going to be red, or is it going to be black, or is it going to be a mixture? Right? Remember, these are on the X chromosome, and according to the lion hypothesis, both of these alleles aren't going to be expressed, right? Only one is going to be expressed in any given cell. for the first few weeks after conception, they both may be expressed, but in by the time the kitten is born, only one of these is going to be expressed in each cell. So cats with this genotype are what we know as calico cats or tortoiseshell cats. And so calico cats and tortoiseshell cats have the same genotypes. And tortoiseshell cats have black patches and red patches. Again, red being orange. And I'm writing kind of small here because what I want to indicate is that the patches are really small. So small that you wouldn't really consider them patches. Calico cats, however, have large patches of black or red or white. Now the white is caused by another gene. So we don't have to worry too much about that, but that's where those white patches are coming from. And the size of the patches, the fact that they're large over here and small over here in tortoiseshell, is due to another gene. It's not due to the coat color gene. So these are similar cats with a similar genotype for the coat color gene. And calico cat is probably easiest to talk about. So if I were to diagram a calico cat here, it would look something like this. And check out the notes because I have some pictures of cats I grabbed from the internet and put in the notes. So if you're not sure what a tortoiseshell is or calico cat is, you can check out those images. And here's my cat here. Actually, I have a cat now and it's orange. It's a female, all orange. So the genotype for my female cat, I'm pretty sure it's going to be CR. Now, this calico cat right here is going to have big patches of black, another patch, let's say, of orange, and let's say a patch of white down here, then another patch of black right here. So these big patches, and that's calico. That's what calico means. So why are all of these black? So they're all black because what? So all the cells have to be this genotype, CR, CB, with one of these inactivated. So in a black patch, we could be pretty confident that all of these cells that are producing the black pigment here have that X chromosome with the CR allele tucked away into the bar body and is inactive. The CB allele, that's the one that's active in all of these cells. And why are they all next to each other? Well, it's probably because, we have a little cell right here towards the middle here, and this is the one that probably at about, let's say, a couple weeks after conception, this is the one, let's say, where the CR allele was inactivated. And every cell that descended from that one as the embryo is continuing to develop has had that same CR, the same X chromosome carrying the CR allele inactivated. Because like the lion model holds, that once an X chromosome is inactivated, every descendant cell has that same X chromosome inactivated. So the large black patches are all black because they're all probably descended from the same cell. And that cell, through that random process, has had the X chromosome carrying the red allele inactivated. Does that make sense? And the same thing for the orange. Somewhere there was a progenitor cell that had the X chromosome carrying the black allele inactivated. In every daughter cell, through every round of replication, DNA replication in cell division, that same chromosome was inactivated. So all of the descendant cells express or have that active X chromosome as the one that contains the red allele of the coat color gene. So the calico cat, tortoiseshell cat, this is an example of the lion model in action. And those colors are related to X chromosome inactivation and the fact that the X chromosome is inactivated during embryogenesis, and once that X chromosome is inactivated, it remains inactive in every daughter cell. Okay, so I hope that lion model makes sense. Okay, so here's a question. So if you see a calico cat, is it male or is it female? Or a tortoiseshell cat, calico cat or a tortoiseshell cat, male or female? Almost always female, almost always female. I'm gonna say almost always female. And I don't know the frequency of this, almost always. But there are rare cases where a cat will be male and calico. And 47 cats with a carrier type. Oh, 47. I don't know how many chromosomes cats have. I can't say that. But what I can say is Klinefelter cats. So these are cats with 2 X chromosomes and a Y chromosome. Now remember, the lion hypothesis holds that if there are more than one X chromosome in a cell, one of them will be inactivated at random. But Klinefelter cats with 2 X chromosomes and a Y chromosome are, the sex is male, but the testes are non-functional, so infertile. They don't make functional sperm. So Klinefelter males, calico cats, or tortoiseshell, are infertile. So, okay, so again, you see a calico cat or a tortoiseshell cat, almost always female, and very rarely you can have a male cat that is calico or tortoiseshell. That's very rare. Okay, so that's it for dosage. Oh no, that's not it. That's not it. Now for the interesting stuff. I mean, I think that stuff is pretty interesting, but even more interesting is the research being done across the world for the last couple decades trying to understand how X chromosomes are inactivated in mammals. So it's very confusing. It's kind of amazing that this can happen. So let's say this is our X chromosome right here. And I'm just going to pick a random location on this diagram of the X chromosome. And we're going to call this area right here XIC. ZIC, I guess it's called ZIC. For X inactivation center. Now the X inactivation center has 4 genes. The whole center, it's about 1 million base pairs long. So 1/150th of the X chromosome, because we saw in a previous video the human X chromosome is 150 million bases long. So 4 genes, one of those genes is called ZIST for X inactive specific transcript. Now, ZIST is an example of a non-coding RNA. What does that mean? So it's not translated into a protein. It doesn't hold the code for a protein. So it's a non-coding RNA. And what it does is it coats the inactive X chromosome. So that chromosome in the barr body, it coats it. Now, why that coating is part of the inactivation process, we don't know. But cyst must continually be transcribed from the inactive chromosome. So the inactive chromosome, cyst is transcribed continuously from the inactive chromosome. So it's sort of, I guess, counterintuitive, right? So most of the genes on the inactive X chromosome are not transcribed, but cyst is transcribed, and then that non-coding RNA coats, or is associated, it covers, let's just say covers, that cyst chromosome in the barr body. Now, before the chromosome is inactive, the cyst RNA is transcribed from both active X chromosomes. And then once one X chromosome is inactivated and the other one remains active, the one that is active and the one that is not put in the barr body stops transcribing cyst, or cyst is no longer transcribed from the active one. So it's weird, huh? It's weird. The X that is put in the barr body is the one that has to keep, from which is the one for where the cyst gene is continuously transcribed. Very strange. So unknowns, so many unknowns with this. So X chromosome counting. Like we said, if in an individual that is 47XXX, somehow cells can determine that there's 3 X's there, and 2 of them have to be inactivated. So how the mechanism that counts the number of X's and keeps inactivating X chromosomes until only 1 is active is unknown. So number 2, how do cells keep same X's inactive? Keep same X. I put X's right here because like we said before, sometimes cells can have 3 X's. How do they keep the same X inactive? Through replication and cell division. Essentially unknown. Essentially unknown. And why? So what is the evolutionary advantage to keeping the same X inactive? Is there an advantage? It seems like, it would be rather difficult to keep the same X inactive, and if it could randomly inactivate just one, that seems like that would be easier. But somehow the cell keeps the same X inactive, and scientists wonder, like, what is the purpose of keeping that same X inactive? There might be some benefit to that we don't know about yet. So that's all we're going to do with X chromosome inactivation. We will probably do more on the mechanism in BSc 350, which is molecular biology. So if you're interested in mechanistic details of phenomena like this, I encourage you to take this course. I'll be teaching this course in fall of 2020, this coming semester. Okay, so I think that's it for lecture 19. I'll see you in lecture 20, where we do sort of a quick introduction to transposable elements. Okay, see you in the next video.